Hello, everyone, and welcome to this journal club. As some of you might know, I'm not feeling too well, so for today's journal club, I decided to recycle 3 case studies that I presented at the World Drug Discovery Forum not long ago. The last case study, as requested by Frank, will be dealing with NAFLD and NASH. So I hope you enjoy, and if you have any questions, please don't hesitate to reach out to me either via email or through Slack. Thank you. So the first study I would like to talk to you about today demonstrates that an untargeted mass spec-based metabolomic and targeted bioanalytical approach can be used together to investigate toxicity of a compound that poses preclinical or clinical developmental challenges in a pharmaceutical setting. The study was published in the journal Toxicology and Applied Pharmacology and was conducted with Merck Research Laboratories. And at the time, Merck was developing a potent nicotinic acid receptor agonist, a compound for the treatment of dyslipidemia. Now, based on exploratory toxicology data in the rat prior to recommendation for development, the compound was known to cause chronic renal failure in the rat. Unexpectedly, post-recommendation, the compound was found to cause rapid onset of morbidity and acute kidney injury in male CD1 mice within 24 hours following administration of a single 100 milligram per kilogram dose of this compound. Subsequently, experiments in a knockout mouse model showed that the renal toxicity in mice was not related to the mechanism of action of this compound, which is called SCH900424, and based on these findings, 2 further investigations were undertaken. Liquid chromatography mass spectrometry and gas chromatography mass spectrometry techniques were used to identify small molecule biomarkers of acute kidney injury that could aid in a better mechanistic understanding of the compound's induced acute kidney injury in mice. The metabolomic study revealed 3-indoxyl sulfate, or 3-IS, as a more sensitive marker of the compound's induced renal toxicity than typical markers such as creatinine and urea. An LC-MS assay for quantitative determination of 3-IS in mouse matrices was also developed. Following treatment with the compound, 3-IS levels were markedly increased in murine plasma and brain, thereby potentially contributing to renal and central nervous system related rapid onset of toxicities. Furthermore, significant decrease in urinary excretion of 3-IS in those animals due to compromised renal function may be in fact associated with the elevation of 3-IS in plasma and the brain. This data suggests that 3-IS has the potential to be a biomarker of renal and central nervous system toxicities during chemically induced acute kidney injury in mice. In addition, based on the metabolomics analysis, other statistically significant plasma markers, including p-cresyl sulfate and tryptophan catabolites such as kynurenate, kynurenine, and 3-indole lactate, were also found to be of toxicological importance. This comprehensive approach that includes untargeted metabolomics and targeted bioanalytical sample analysis could be used to investigate toxicity of other compounds that pose preclinical or clinical developmental challenges in a pharmaceutical discovery and developmental setting. The second case study I wanted to mention today is a study published in Cell. The title of the publication is Microbiota Modulate Behavioral and Physiological Abnormalities Associated with Neurodevelopmental Disorders. Now, this study stems from the observation that neurodevelopmental disorders, including autism spectrum disorder, are defined by core behavioral impairments. However, subsets of individuals display a spectrum of gastrointestinal abnormalities as well. However, in this case, how the microbiome produces this effect and also how to monitor it and the translational potential was unclear. And so in the study, the investigators decided to focus on this. And first of all, the researchers at Caltech were able to demonstrate gastrointestinal barrier defects and microbiota alterations in the mature immune activation mouse model that is known to display features of ASD. They found that oral treatment of maternal immune activation mouse model offspring with the human commensal Bacteroides fragilis corrected gut permeability, altered microbial composition, and ameliorated defects in communicative, stereotypic, anxiety-like, and sensory motor behaviors. So the MIA offspring display an altered serum metabolic profile, and Bacteroides fragilis modulates levels of several metabolites. In fact, treating naive mice with a metabolite that is increased by MIA and restored by Bacteroides fragilis causes certain behavioral abnormalities, suggesting that the gut bacterial effects on the host metabolome impact behavior. The translational potential of this work was demonstrated by 4EPS being confirmed in a human cohort. And taken together, these findings support a gut microbiome connection in a mouse model of ASD and identify a potential probiotic therapy for gastrointestinal and particular behavioral symptoms in human neurodevelopmental disorders. Amongst the findings that are actively being pursued, a translational biomarker, that is 4EPS, has become one of the cornerstones to developing and designing and monitoring therapies directed against dysbiosis-driven autism. Now, as I mentioned, the last case study I am going to present will be touching on non-alcoholic fatty liver disease. So I wanted to give you a bit of key facts just before jumping into the case study. Now, for those of you who don't know, the liver is the powerhouse of the body and has multiple functions, including the production of clotting factors, sugars, fat, but also it metabolizes drugs. Now, non-alcoholic fatty liver disease, or NAFLD, affects 1 in 3 adults and 1 in 10 children in the US. It is so strongly associated to obesity and diabetes and can lead to fat accumulation in liver cells. And this is called steatosis. With non-alcoholic steatohepatitis, which is NASH, it's like a much more severe and chronic condition where there is an inflammation and damage to liver cells that may lead to scarring of the liver or fibrosis. Now, fatty liver is a silent condition. There are no symptoms until the patients develop advanced liver disease or cirrhosis. And simple blood tests or ultrasound may suggest fatty liver disease, but diagnosis of fibrosis and NASH do require a liver biopsy. So just before jumping into the case study, again, a few more key facts. The study I will be presenting is a phase 2 randomized double-blind placebo-controlled clinical study. And it led to a publication called GS0976, Reduces Hepatic Steatosis and Fibrosis Markers in Patients with Non-Alcoholic Fatty Liver Disease. It was published in Gastroenterology in November 2018, so it's fairly recent. And obviously, it stemmed from a collaboration that Metabolon had with Gilead. I hope you enjoy. Finally, the third and final case study I would like to talk to you about today is a study we did with Gilead. It was published in the journal Gastroenterology, and the title of the publication is GS0976 Reduces Hepatic Steatosis and Fibrosis Markers in Patients with Non-Alcoholic Fatty Liver Disease. And obviously, the study was conducted whilst developing a drug for non-alcoholic steatohepatitis, for NASH, And NASH is characterized by the presence of hepatic steatosis, inflammation, hepatocyte ballooning, and variable degrees of fibrosis in patients who do not consume excess alcohols. Patients with advanced fibrosis have the greatest risk of liver-related morbidity and mortality, attributable to hepatic decompensation and hepatocellular carcinoma. A central feature of NASH is the aberrant regulation of lipids within the hepatocytes. In fact, increased endogenous fatty acid biosynthesis, or de novo lipogenesis, impaired fatty acid oxidation, the generation of biologically active fatty acid signaling molecules, are all important factors in NASH pathogenesis. In particular, the regulation of de novo lipogenesis plays a central role in fatty acid synthesis and catabolism. The rate-limiting step of de novo lipogenesis is the conversion of acetyl coenzyme A to malonyl-CoA by the enzyme acetyl-CoA carboxylase, or ACC. Now, ACC has two isoforms which have distinct cellular distributions and hence unique functional roles in fatty acid metabolism. Now, since inhibition of ACC1 and ACC2 isoforms would be expected to reduce de novo lipogenesis and enhance mitochondrial beta oxidation, respectively, these complementary roles support ACC inhibition as a potentially attractive therapeutic target in NASH. And at the time, Gilead was evaluating GS-0976, and it was liver targeted. It was a small molecule allosteric inhibitor of both ACC1 and ACC2 in development for the treatment of NASH. And in preclinical models of diet and genetically induced obesity, ACC blockade had been shown to decrease hepatic steatosis, inflammation, and insulin resistance. Again, in an open-label proof-of-concept trial, 10 NASH patients with GS-0976 20 milligrams daily for 12 weeks had a median decrease of 29% in hepatic de novo lipogenesis and significant reductions in hepatic fat, liver stiffness, and serum tissue inhibitor of metalloproteinase 1, another marker of fibrogenesis. So very importantly, this study aimed to evaluate the safety and efficacy of GS-0976, as I said, an inhibitor of acetyl-CoA carboxylase in liver. In phase 2, randomized placebo-controlled trial of patients with NASH was conducted. The study included measures of hepatic steatosis, stiffness, serum markers of fibrosis, and plasma metabolomics as well. Now, since the relative abundance of peripheral acylcarnitine species reflect the efficiency of mitochondrial beta oxidation, an unbiased plasma-based metabolomic screen was performed to identify changes relative to ACC inhibition with GS-0976 treatment. Patients were categorized across treatment groups and according to MRI and MRI-PDFF response. Although minimal differences were observed between treatment groups, multiple long and medium chain acylcarnitine species were significantly suppressed in GS-0976 20 milligram treated group who achieved a PDFF response versus those without a PDFF response. On the contrary, patients in the GS-0976 5 milligram or placebo groups showed minimal changes in acylcarnitine species when compared to PDFF response. This data supported measurement of plasma acylcarnitines as potential peripheral markers of improved fatty acid catabolism with ACC inhibition by GS-0976. So ultimately, this study was able to show how a panel of metabolites are promising early biomarkers of imaging response in NASH patients, providing a mechanistic link between acetyl-CoA carboxylase inhibition and effects on liver fat content, as well as increased program confidence in this successful phase 2 trial. And ultimately, it also showed how biomarkers can accompany the future development for reading out the pharmacodynamic activity and response of